Hi everyone. My name is Hunter Ryder, and I'm an undergraduate researcher at the University of West Florida. I'm currently working in Dr. Alexis Janicic's research lab to utilize the molecular tool of environmental DNA to detect specimens of the river redhorse in the Escambia River. The river redhorse is a suckerfish of the Catostomidae family, and it's characterized as using large lips and molar-like teeth on the pharyngeal arches to vacuum and consume invertebrates from the substrates of streams and lakes. The river redhorse inhabits pools and runs of small to large rivers and is usually found over gravel or mixed sand and gravel. It is a late spawning fish that spawns in the middle of April when water temperatures are around 22 to 24 degrees Celsius. The river redhorse is not an economically exploited species, but it is ecologically important. This species facilitates the transfer of matter and energy from the benthic food web to the pelagic food web at the ecological level. The river redhorse is one of 6 North American fish that can eat invasive zebra mussels and is suggested as a suitable species for biocontrol of invasive mollusks. In Florida, the river redhorse is only found in the Escambia River, making it a distributionally isolated population. It is listed as a species of greatest conservation needs in the state of Florida due to drastic population decline in large parts of its native range. The river redhorse has been recorded as collected only 3 times in the past 60 years, with the most recent collection being in 2017. This is in part due to habitat alteration, such as channelization, impoundment, turbidity, siltation, and other forms of pollution. These threats act as limiting factors because river redhorse are inflexible in their habitat requirements and are intolerant of pollution and heavy siltation. The Escambia River and other bodies of water have long been polluted by industrial plants, landfills, and septic tanks here in Florida. Multiple tributaries in the mouth of the Escambia River are not held to proper water standards by the Florida Department of Environmental Protection. Food resources of river redhorse are affected and reduced by these toxicants, which in turn reduces populations of the river redhorse. The river redhorse population in the Escambia River is likely being affected due to the pollutants in the water, which cause reduction and extirpation of invertebrates, the food source for this fish. Because of the rarity of catching river redhorse and the time and money needed for conventional sampling methods, an alternate approach is ideal. Environmental DNA is a relatively new tool that has been used to detect the presence of aquatic species. Environmental DNA consists of sloughed-off skin cells, intestinal cells, scales, and/or mucus of aquatic species that can be extracted from collected water samples. The rapid degradation time in freshwater ecosystems makes environmental DNA very useful for detection of species for conservation purposes. A positive detection is likely to be associated with the current presence of species and populations. The process of using eDNA is also less destructive to habitats compared to traditional methods of sampling, and it also allows for the detection of the elusive and/or rare species without handling specimens, which can cause stress and/or mortality. Sampling for water collection will start at the mouth of the Escambia River and continue up past the Alabama-Florida border. The full length of the Escambia River, as well as the lower Conecuh River in Alabama, will be sampled. The lower Conecuh River will be sampled because a small population of river redhorse is thought here and in Murder Creek, a tributary of the Conecuh. Collection dates will be monthly, beginning in December and ending in May, to determine the locations of the river redhorse before, during, and after their spawning migration upriver in the month of April. 3 15-milliliter water samples will be collected from the surface at each determined location, and the water will be preserved in 1.5 milliliters of sodium acetate and 33.5 milliliters of ethanol. The samples will be placed on ice to protect the environmental DNA from damage and/or heat exposure while out in the field. Water samples will be transported to the Janicic lab at the University of West Florida after collection, and DNA will be extracted using a Kigen DNA Easy Kit. Extracted DNA will be amplified using species-specific primers of the river redhorse and polymerase chain reaction. Amplified DNA will then be separated into fragments by gel electrophoresis in order to determine a positive or negative detection of the river redhorse at a specific location. Positive detections will be sequenced bidirectionally to confirm eDNA detection. The anticipated outcomes of performing these methods are to determine the presence of the river redhorse and where it is located in the Escambia River. Positive detections can give insight on where to perform conventional methods of sampling in hopes of catching a specimen for further conservation on the population of the Escambia River, as well as limiting any habitat damage. Known locations of the river redhorse will also allow for the population's habitat and spawning routes to be managed in order to restore and preserve river redhorse and other species in aquatic habitats in Florida.